Okay, then welcome everybody, and it's an honor for me to present some of the research we've been doing in our group over the years, and I'm going to present more the methods and the approaches we are using in the group with some examples, and also some examples from Argentina, but not exclusively. Just to give you an impression what we understand under Earth surface geochemistry. This is of course work I don't do alone, so there's a lot of people involved. I present examples from PhD students and also from other collaborators. So organic surface geochemistry is a word, but the question is what do we understand under this, and the research we do here is focused on two main topics. That's first the understanding paleoclimate, understanding the effects of climate change on the hydrological cycle as one major topic. And I'm going to present some examples of that, how we looked at interactions between tectonic processes and regional hydrological changes in the northwest Argentinian Andes. And also an example from Europe where we look at late glacial hydrological changes. And a second major research topic in our group is we aim to look at the role of organic carbon in the global carbon cycle over longer geological timescales. So what are the processes which export carbon, which oxidize it, or which bury it and remove it from the carbon cycle for geological time periods. Because this is important to understand how and why Earth's climate is relatively stable over geological timescales. With stable here, I mean that it's stable enough to sustain, to develop, that life could develop and actually could be sustained. So this is looking at longer geological timescales. This is related to the project we are planning to do within strategy as well. And I'm going to present something about this at the end of the talk. The methods we are using are geochemical methods, organic geochemical methods, and that combines all these different research questions. And I will also give you an example about this. But first of all, I want to talk about the motivation, why we are interested in these kinds of questions in the hydrological cycle, as well as in the carbon cycle, which can be, of course, driven or forced by the hydrological cycle. So this picture shows, it's taken from the IPCC report, from the latest IPCC report. It presents multi-model averages of predictions of climate in the year 2100 under in this case under an emission scenario which is equivalent to 1,200 ppm of CO2. So what these models, what you can see here, this is temperature and this is precipitation. What you can see on the upper panel is that all the models predict that it's going to be warmer everywhere on the planet. So that is pretty well known, although there are some regional differences. But also what you can see in these stippled areas is that all the models, basically, or 90% of these 35 or so models, they agree on the predicted sign of change. So that means we can be pretty sure that under higher CO2 concentrations we also really get a warmer climate. But if we are looking at the lower panel, this shows how the same models predict precipitation changes. You see quite a lot of differences. First of all, the response is not linear, it's not unidirectional, so some regions are predicted to become wetter or drier, like these yellow colors. Others are predicted to become wetter, so there will be quite a regional difference in the response of the climate system to this. And so it really depends on where you live, how precipitation change are going to affect you. The other point is that this is the areas I've highlighted here in red. This is where the models actually are uncertain if there's any change or they are not agree on the sign of change. Some models predict it's going to be wetter, others are drier at the same place. So that means that some of the underlying processes which are going to affect or which are affecting the hydrological cycle responding to climate change are not really well understood yet. So what we can do, of course, is make these models better, but since I'm a geochemist, what I can do, I can look into the past and try to understand how past climate change was forced and how the regional development of this was. This is not only important for understanding paleoclimate, but it's also important for understanding the response of the carbon cycle, because rivers, for example, transport a lot of carbon from the terrestrial environment into the oceans and then remove it possibly from the short-term carbon cycle. So all these effects are connected to the water cycle, and that's why one of the things we are really interested in here is understanding the processes in the hydrological cycle. One very useful tool to look at the hydrological cycle are the stable isotopes in water. And I'm sure you all have heard about, know what stable isotopes are. What you can see here on this graph is that this is the isotopic composition of precipitation in the world, shown as a delta D value. So that is basically the ratio between deuterium, the heavy isotope, and hydrogen, the lighter isotope in water. So the blue colors represent negative delta D values, so that means that you have less deuterium than in seawater, which is used as a standard, and the positive values are regions where you have more deuterium than seawater, which is the standard. So by just looking at the colors, you can see that there's a lot of climatic and hydrological information contained in these isotope ratios. One obvious effect is temperature. So if you go to the cooler regions of the Earth, there is much more deuterium depleted precipitation, or for example here. If you go to the driest places on Earth, you see that they are actually very enriched in deuterium precipitation there. This is because of evaporation, because the lighter or the water molecules with the lighter isotope preferentially go into the vapor phase, which leaves the residual water enriched in the heavy isotope. Another important effect is the so-called continental effect. So you can basically track the pathway of the moisture. For example, in our region, we get the moisture mostly from the Atlantic Ocean, and that starts to rain out when it reaches Europe, and then it travels further onto the continent. So since the water molecules with the heavy isotope rain out first here on the coast, the precipitation there is more enriched in deuterium. The further we go into the continent, the more depleted this moisture gets in deuterium because it has lost it already, and precipitation falling more within the continent is more deuterium depleted. So hydrogen or also oxygen isotopes in precipitation are a useful tracer for hydrological processes today, but we can also use geological archives which record this isotope ratio to look at the past, for example. You probably are familiar with carbonates as a tool for this, carbonates and oxygen isotopes in carbonates or in ostracods or in certain shells. But the problem is that not every geological archive contains carbonates or these shells. So what we are focusing on is organic material, because in almost any geological archive we can find organic material. Because organic material is synthesized through photosynthesis. So you take water, CO2 and energy, and then you produce organic molecules and oxygen. And these organic molecules are, for example, cellulose, but also lipids, and this is what we are looking at, because these lipids are also stable in the geological record. Cellulose is of course more degradable, but you may be familiar with this cellulose and studies, for example, tree rings. But if you want to go further back into the geological time, then there is not much cellulose left, so we can look at these lipids, and we call these compounds biomarkers or molecular fossils, because there are certain of these lipids, of these organic molecules, which we can trace to their biological sources. For example, this compound, this is a long chain hydrocarbon. This consists of of carbon and hydrogen atoms, and it's an N-alkane with 29 carbon atoms in this case. This is a compound which is synthesized in the leaf waxes of higher terrestrial plants. So the shiny layer here on top of the leaves, this is the wax layer which protects the plant from evaporation, but also from physical processes. And this is mainly made up of compounds like these. Yeah, and these compounds are stable in the geological record. If you look millions of years in the past, we can still find these compounds. And these long chain compounds, we only find them in the leaf waxes of terrestrial plants. It's not that we can say what tree species was present at that time, because a variety of trees produce the same compounds, but we can at least say this was terrestrial vegetation. If you look at other compounds, for example, this shorter chain hydrocarbon, or this with C20 with 23 carbon atoms, so these are characteristic for aquatic plants or even for bacteria. The shorter the chain gets. There are other types of biomarkers which are listed here, like fatty acids or certain sterols, for example, or hopanes, which are characteristic of bacteria. So there's a variety of these compounds which you can identify in the geological record, which we can then use to understand where the organic material comes from. Was this terrestrial plants, algae, or even in some cases we can recognize a specific type of algae, for example. Another advantage of these compounds for our analysis is, because we are interested in the isotopic composition of these, which I'm going to tell you in a minute, is that they consist mainly of carbon and hydrogen, and the bond here is very stable. So that means we have no isotopic exchange when this material is in the sediments, as long as it's not heated up to a certain degree, like 100 degrees. So if the material is not matured, then we can still determine the original isotopic composition of these compounds. By the way, if there are any questions, feel free to interrupt me, because I don't know what your background necessarily is. So I may tell you things you already know, or I may just not explain things enough. So just let me know. So the way we extract this, we have to get this material out of the sediments to analyze it. That's a lot of lab work. And we basically extract the material with organic solvents from the sediment, for example. And then we have to separate these different compound classes into different fractions. And we do this on these silica gel columns, where the brown stuff, this is the extract, and this is then flushed with different types of solvents, and we collect, so these solvents dissolve a certain fraction of these organic molecules according to their polarity, and we collect these in different fractions to purify them, to be able to analyze them on a machine which looks like this. So this is a gas chromatograph coupled to an isotope ratio mass spectrometer, and we get this chromatogram. This after the compounds are injected here, vaporized, and then separated on a capillary column, and every compound has a different retention time. That's how we call the time that needs to get to the detector. So the compounds with more carbon atoms, this is indicated by the black numbers, they are, for example, they are remaining longer on this column. So that's why we get this nice separation according to their chain length in a gas chromatograph, so that we find these C29, the long chain hydrocarbons, the leaf waxes here in the later part of the chromatogram, whereas the shorter chain compounds like this C23 alkane, which comes from aquatic macrophytes, we find this in the first part of the chromatogram. And then, since it's coupled to an isotope ratio mass spectrometer, we can analyze these peaks, and we get isotope ratios here expressed as delta D values for every of these individual peaks. So that's a lot of lab work to be able to clean them and to do the analysis, but this is basically how we can analyze them in the lab. So, but when we measure the isotopic composition of these compounds, what does it actually tell us? Well, we probably you think that this is related to the isotopic composition of the precipitation, since I've been talking about this before, and yes, it is, and we can also test this. So what we have done in the group, for example, is to look at hydrological gradients. So this is an example from Cameroon, which is here indicated on the map of Africa. And Cameroon is actually a country which is characterized by very strong precipitation gradients. So in the south here at the coast, this is basically a tropical rainforest, and then the further you go up in the north, it's getting much drier. And you can also see that here in the relative humidity. So what we have done there, and this is work by Yannick Garçon, who is also at the University of Potsdam postdoc, is looking at lakes which are distributed along this gradient and analyze the sediments in these lakes for these biomarkers, for example. And this is a lake example from this rainforest lake. This has a precipitation of 5 meters a year, and this is in the north where you have a precipitation of less than 300 millimeters a year. So it's a very strong gradient, and so what we can then do is to compare the isotopic composition of precipitation in this location with the isotopic composition of the C29 leaf wax analkane again. And this is the data from the Cameroon transect, and these are the blue dots are data from a European transect, which I did in my PhD. And so you can see that there's a really nice linear relationship between the isotopic composition of precipitation and the isotopic composition of that leaf wax. You note that the composition here is much more depleted in deuterium, much more negative values than from precipitation. This is because there is a biosynthetic fractionation during the uptake of the hydrogen in the plant. This is same for carbon, because plants or any photosynthetic organism, they prefer to take up the lighter isotopologues of the water. So there's always a fractionation involved. That's why we get this offset between these and these values, but we still find a really nice linear correlation, which even works almost on the scale of a whole hemisphere, where the lakes are quite different in terms of their trophic state, or, for example, what organisms are living in the lake. So it seems to be quite robust, and there is a lot of data in the meantime from other lakes from all over the world, and all of these data points fall along this linear regression. The same is true for other compounds, like again this mid-chain analkane. This is actually data from actual water plants and sediments from Tibet in that case. So it seems to be very robustly recording the isotopic composition of precipitation. So we can really use this as a tracer for this in the past, for example. But if you look really closely, you see that the relationship here is, although it's quite well constrained, it's quite a good correlation, but it's not a one-to-one relationship, so the slope here is much less than 1. So that indicates that maybe precipitation is not the only factor influencing this. And it makes a lot of sense, because in the way we are, or maybe go back a second, but if you look at this regression line for these water plants, this is actually much closer to a one-to-one relationship. So what's the difference between water plants and terrestrial plants? So we have to think about the way how plants are taking up water. So they are, of course, not taking the precipitation water directly. So plants are taking water through the soil, where you can have evaporation in the soil. So that would enrich the isotopic composition of the water the plant's taking up relative to the water. Although in a very densely forested area, this is not a strong effect. But when the plant is taking up the water and transferring it to the site of photosynthesis, which are the leaves, then there is another fractionation process, because the plant is losing water to transpiration. And that, again, so the leaf water in the plant, so the water which is in the leaf, which is the source of hydrogen for biosynthesis, this water becomes enriched in deuterium because of plant transpiration. And this enrichment gets stronger if the drier it gets. So in very dry areas, you would actually expect an additional enrichment of the leaf water, and then also the leaf waxes in these compounds. So if you put this into a scheme, and this may be a bit complicated, but it's important to understand this for the later part of the talk, we can think about this as this is the water a plant is taking up. And let's think of a water plant which lives in this water. It just has a certain biosynthetic isotopic fractionation and produces lipids with a more depleted delta D value. But it more or less directly records the isotopic composition of the water in the lake where this water plant has lived. If we think about a tree standing next to that lake, which receives precipitation water, and maybe a little bit of soil water evaporation, but usually that's not very significant. But then the water in the leaf is enriched in hydrogen, and from this water biosynthesis starts and creates lipids which are depleted in deuterium. So what a leaf wax analkane delta D actually tells us is something about precipitation, but also about transpirations, about evapotranspirational processes. So these two factors are in there, and it may sound like a complication because we have these two effects. if we can disentangle these effects, I'm going to show some examples later, we can actually get information about past evapotranspiration from geological records. That was more or less the introduction about the methods and about the proxy. So if there are any questions about this, I'm happy to answer them now, because otherwise I would go on and tell you some examples of how this conceptual framework can be used to look into the past. Questions? Oh, okay. Then let's go to Argentina and to northwestern Argentina, probably the area which you have at least partly visited. Have you been to Angastaco Basin? No. Okay, so I'm going to show then some data from this Angastaco Basin, where this is work by Alexander Rohrmann, who was, I think, also on the trip. And he has done this together with Manfred and me. Part of his PhD thesis was to look at a past record of hydrology and relate this to tectonic processes, the uplift of these intramontane orographic ranges building the intramontane basins. And he was using these leaf waxes as a tool. I'm going to show you some of his results. So we're looking at this Angastaco Basin, and since you've been there, I don't really have to tell much about the area. It's one of these intramontane basins, and it contains a very thick sedimentary sequence of fluvial and lacustrine deposits. So it's about, I think if you've been there, you know it's a spectacular sight to see this, and you can walk past these sequences and walk back and walk back in time, for example. And this is like almost more than 2 kilometer sequence. He has looked at the Palo Pintado and the San Felipe formation. And the green dots are samples he took for biomarker analysis, and the yellow dots are samples he took for soil carbonate analysis, because we find abundant of these soil deposits. So that sequence covers a time period from about 2 to almost 9 million years. And this is also the time, during that time, we think that the orographic barrier actually became active and this intramontane basin developed. So this is now a plot which shows the isotopic composition of leaf waxes. Here this is the C29 and the C31 N-alkane, so that's the blue line, compared to the isotopic composition, oxygen isotopic composition of soil carbonates over this time period from 9 to 2 million years. And what you can see here is that there are substantial changes, and that on the first order these two signals look similar, but they are not exactly similar, and I'm going to tell you in a minute also why. But we can see substantial changes in the hydrological development of these deposits. Interestingly, we see a decline here, then more or less some variability, but more or less, let's say, the same range of values until we get an enrichment of values and then remain at relatively high values, which, of course, there's less of a temporal coverage here, but they seem to be more stable also these values during this time period. So wetter and drier, but that's what we could say at these points in time. And now if we think about the tectonics, actually, we can see that during this time, during around 6 to 6.5 million years, when we get this aridification or the dryness, this coincides actually with the development of the orographic barrier. So that it indicates the orographic barrier came up, and in a time period of, let's say, a few 100,000 of years. And then the basin became from really wet, and we find also evidence in a lot of fossils that this has been quite a wet basin. You may even have seen some when you were in the field. But then afterwards we get this aridification in the basin. So the leaf waxes and also the soil carbonates, they seem to record the history of this basin development quite nicely here. So basically we transition here from a foreland stage where we have wet conditions with abundant fossils indicating also rainforest type taxa at some point. We transition into this intermontane basin phase, which is significantly drier in that case. So based on Alexander's work, he could first of all say that the hydrological cycle in this specific region, in this Angastaco basin, has changed actually quite rapidly as a response to topographic growth. This is because if you reach a certain uplift or a certain altitude, then your range becomes an orographic barrier. This is today at around, I think, 2.5 kilometers or 2.5 kilometers when this actually becomes a barrier. And then, of course, the hydrology within the basin changes quite fast. And with fast here, I mean on 100,000 year timescales. I'm going to talk about faster processes on decadal timescales later. But for now, this is the fast process. And that, of course, the uplift of that basin has controlled the moisture supply, and the vegetation has adapted also to this process. And I didn't talk much about evapotranspiration, but because I'm going to talk about this in a minute. But this, when we use the different isotopes from the soil carbonates and the leaf waxes, we can also look at the difference between those and look at evapotranspirational changes, which Alex has done in his thesis. But I'm not going to go into detail on that. But I'm going to show you another example now, which is located here in Europe, and we're going to look at a much shorter time scale just to see what we can do with this proxy before we can go back into Argentina. Because I think it gives you a nice example of how precise this proxy actually can be. So what we're going to look at now is at hydrological changes at the end of the late glacial period here in Europe. I want to compare what happened in Greenland, recorded in the Greenland ice core record, with a record we've obtained from Lake Meerfelder Maar, which is in the Eifel region in western Germany. So this late glacial period is actually quite an interesting period. You can see here the isotope record of the Greenland ice core, which shows us this, coming out of the ice age. Then we have the warming period during the Berling-Allerrod, and then we have another period of 1,000 year duration where it became significantly colder and very abruptly so. So this is on the order of 120 years, where we get a temperature drop of about 4 degrees in Western Europe. And then with the onset of the Holocene, which is located here, we get much more stable conditions until we come to today. So this period of what's sometimes called the Younger Dryas period, in the ice core is called Greenland Stadial 1. This is the last major abrupt climate change we had here on our planet. So it's a nice example if we want to understand mechanisms of abrupt climate change, because we have abundant records and we have a pretty good understanding of what happened at that point in time. The Greenland ice cores, you know about those, I guess, but there's also, but of course the problem with the ice cores is they are in Greenland or in the polar regions of the Earth where no one is living. But if we're interested in how regional climate and especially regional hydrology is going to respond to future climate change, we should look at places where we live, in our backyards, for example, and we have these beautiful lake sediments which exist in some of the Eifel Maar lakes, but also in other lakes, which are annually laminated. So every of these layers corresponds to a year, so that we can actually place this boundary. This is the boundary between the Allerrod and the Younger Dryas at a time of 12,679 years before present, because you can count these layers, and you can additionally compare it with tephra eruptions which are recorded, or volcanic eruptions which are recorded by tephra layers in these lake sediments, like the La Hazé eruption, which happened 12,880 years before present. So we have relative and absolute, very well constrained absolute time markers, so we have a really good control on the chronology. The nice thing is also that these lake sediments contain abundant of our lipid biomarkers, again our long-chain analkanes, which in the case of Meerfelder Maar, which is mainly produced by birch or salix or grasses, which again, as I told you, records the isotopic composition of the leaf water. But we also find evidence from biomarkers of submerged macrophytes, in this case potamargeton, which would then record the isotopic composition of our lake water. So this is how this water plant looks like. You may have seen it. It's very abundant in lakes all over the world. So if you look at this now, the isotopic composition of these two lipid biomarkers, here the blue line is the aquatic lipid and the green line is the leaf wax lipid. If we look at how this changes during the Younger Dryas period in this Lake Meerfelder Maar, and we compare it to the Greenland ice core record, this is shown here, we see first of all that these two curves look actually quite similar. If you look at the blue curve for now, we have a drop of values going into the Younger Dryas, more depleted because it got much cooler. And then at the end, when we'll be warming going into the Holocene, we get a rise in values. So that actually looks pretty similar if you look at the ice core. So we have a similar signal recorded here in Europe as we have in the ice core. Can we be really sure that this is also from the same time period? Well, yes, we can, because there is a tephra layer from an Icelandic eruption, the so-called Vedde ash. This is a very prominent ash layer in the ice core record, but it's also present. We find this in little traces also in the Meerfelder Maar record. And so, since both of these are laminated, we can actually compare now the differences between the records on decadal timescale. So we can really look at leads and lags in the climate system over decades only. And if we look now in detail, while these curves are similar, there's also quite a number of differences. First of all, we look at this purple line. This is the thickness of these annual layers of these wharfs in the Meerfelder Maar sediment. This is used together with changes in vegetation, which happened at that time, to define the onset of the Younger Dryas. So that's in the lake. So that's this brown, gray box, which I show here. So the increase in wharf thickness happens at a time when in Greenland the change is already more or less done. So there's they're at the lowest values, but this is when at Meerfelder Maar we are seeing the major vegetation changes, and we also see the sedimentary changes. But our isotope record actually looks, we see these changes in isotopes already at the same time as they happen in Greenland. So that records the temperature most likely. So we see that cooling in Greenland and cooling in Central Europe or Western Europe were more or less simultaneously. Yeah, so the cooling happened at the same time, but why did it take 170 years from the onset of cooling to the onset of these sedimentary changes as well as vegetation changes, which we know existed from pollen? To look at this, we're going to look at another proxy. You can see that while these curves are similar, they're not the same. You can see the overall pattern is the same, but there are differences between these two curves, which makes a lot of sense because I explained to you what these actually record. So the terrestrial biomarkers, they should record a evapotranspiration signal, whereas the aquatic biomarkers should not. So what we can do now is we can, coming back to this scheme, we can define a proxy which we call epsilon, terrestrial aquatic, and if all these biosynthetic fractionation processes are constant, which we can assume that this is the case, then this epsilon terrestrial aquatic should be related to the enrichment here. So it should be related to the transpirational enrichment in the leaf water, which is controlled by relative humidity. So we call this our proxy for evapotranspiration. So if we plot this here, and this is the brown line now, you can see that there are more or less stable values in the alarids and variability, but then we get this increase at the same time when we get the increase in the sedimentary, the change in sediment, in the structure of the sediments, but also in the change of vegetation in the pollen record. Yeah, so this indicates that we have much we have higher values, so that indicates much drier conditions at the onset of the Younger Dryas, and that these drier conditions were actually then responsible for changing the environment at the lake, more so than temperature. So temperature, of course, was declining already before, but the ecosystem there in Western Europe or around the lake, that changed because it got drier at the onset of the Younger Dryas. The Younger Dryas was a drier period. We know that because we see that in the pollen record, that there are plants which are not only more adapted to colder, but also to drier conditions. But how can we explain this? Is this also possible? And can we explain this 170-year delay which we observe between cooling and the onset of these drier conditions by some process. We can, so what we think what happened is that the Younger Dryas, as you may know, there are different hypotheses, but one is that it was forced by input of fresh water into the North Atlantic because an ice dam broke on the Laurentide ice sheet, that a lot of fresh water got into the North Atlantic, and this basically disrupted the thermohaline circulation, bringing much less warm air into Europe. So it got colder in Europe, and then you have, of course, sea ice in the winter is much more expanded to the south. So there are reconstructions from ice rafted debris which show that during the mid-Younger Dryas, for example, we had sea ice as far as Ireland, but during the maximum conditions probably as far as southern Spain, northern Spain, southern France. That of course also means that the polar front, so the high pressure system here over that ice, is also expanded to the south, and this probably pushed the whole west wind system, which brings our weather today, much further to the south, and channeled cold and dry air into Western Europe. And this is the location of Mer Ferdinand, which really lies within that storm track, so that the westerly wind system was much more to the south, and that cold and dry air came into Western Europe. And this is also a mechanism how we can explain this delay of 170 years, because if you can imagine, if it gets cooler, you need some time to build up that ice sheet until it was not an annual ice sheet, but a winter ice sheet, that it comes as far back south to produce the patterns we observe in these paleo records. So to maybe summarize this, I think I showed you a few examples that biomarker isotope analysis can be a powerful tool to look at the evolution of long and also short-term changes in the hydrological cycle from geological archives. The results of Alex's work suggest that in the Miocene, when we have the rise of the orographic barriers there, that the hydrological cycle changed on a rapid, in this case 100,000 year timescales, and these dry intermontane basins have developed. And in the last example I showed you on a much shorter timescales that we can really explain this delay between cooling in Greenland and onset of drier conditions in Western Europe by looking using these highly resolved archives and these lipid biomarker proxies. To go back to South America and show you another example of how we are using these leaf waxes, we first have to take a small detour through the Himalayas, and because I want to show you some data from a study we conducted in the Himalayas. As I showed you before, so this is the same plot I've shown you before, the isotopic composition of the leaf wax is related to the isotopic composition of the water. But these data now are not from lakes, now they're from river catchments. So these are small rivers which contribute to the Arun River, which is a major trans-Himalayan river. And since we have an altitudinal gradient, which you can see here, is data plotted against elevation, see that leaf waxes which are exported from catchments in the higher in the higher regions, they are more depleted in hydrogen because it's in deuterium because it's cooler there, for example, compared to the lower latitudes. So we can not only use these as a tracer for wetter, drier conditions, we can also use those as a tracer for the origin of these compounds. So these leaf waxes transported in these rivers have basically a distinct isotopic signature, which you can use to trace their actual origin. Why is this important? Because it's important because we are looking at a system in northwestern Argentina, the Rio Bermejo, which is a mountain, it's a river draining the northwest Argentinian Andes. So this is, I think Salta is a little here, so this is Oran, and this is the Puna Plateau. So that river drains the part of Bolivia, of the Bolivian part of Bolivia, but also part of the San Francisco River catchment and the Humahuaca Basin, where I think you also have visited. So this river transports a lot of organic material because it exports a lot of sediments, as you can see here. It actually looks like pretty brown. transports that through the lowlands, and which at some point goes into the Rio de la Plata river. So there's a lot of material which is being exported from these mountain ranges, and we're interested in where is this material coming from, and why, what happens with the material on the way. Yeah? Because if you remove carbon from modern carbon from a soil and you bury that into the ocean for some time, you remove carbon from the carbon cycle for a significant period of time, which may provide some imbalance in the global carbon cycle. And we are interested in understanding the processes which are actually responsible for this transport mechanisms. So in this case, we want to use these leaf waxes, and I cannot show you data because this is part of the strategy project which we are going to start soon. But I'm going to show you some of the preliminary data we have obtained on some field trips. So if you go along that river, this is how it looks like when you exit the mountains. This is when you, so this is then at 8 kilometers, and this is further, basically going further down the river until it actually flows into the Rio Paraguay, which continues onwards to Buenos Aires at some point. You see it looks similar, but also different. Especially when this is still you're in the foothills of the Andes, so you have mountains. But if you look at this region here, that looks quite different because the river has incised here by like 10 meters into its own sediments, and this is actually related to the forebulge of the Andes because you have developed this tectonic expression on the surface of about like 10 meters, which is about 300 kilometers away from the foothills. Why is this interesting? This is interesting because when you are in the field and you see things like this, you see that there's whole trees and these whole soil profiles, they're falling into the river. So the river actually erodes the sediment in this region where it's actually incising in its sediment, and there must be a lot of material, including carbon, must be added at this particular location. So what we have done, we have analyzed the water chemistry so far. So these are the different stations. This is the San Francisco River, and in the background I've shown you again this profile. So the forebulge region would be somewhere here, and what you can see without looking at the details, that the chemistry of that river water is, this is a tributary, so basically you have to look from zero. It's not changing much, then it's changing here in this area, and then it's constant. Same thing happens here. So the major change happens in this region of the forebulge again. Yeah? So this is CAT and anion data. So this indicates again that there's something getting into the river in this forebulge region, this tectonic expression. So this may be quite an important region for the export of carbon, for example, and that's what we're interested in looking at in this project. We also see evidence for changes in the stable isotopic groups, stable isotopic composition of the river water, exactly happening in the forebulge region. So there must be also some groundwater contribution possibly coming in from the sites. So the next steps, so the initial hypothesis we have developed here is that the geomorphic surface expression of tectonics of the forebulge, that this is actually responsible for adding a lot of material into the river, and including possibly organic material. And this is what we are going to look at with a variety of our biomarker and also other organic mass spectrometric methods. So this is more or less an outlook, and this is what we hope to do very soon with a PhD student in our strategy project, so that we can go back to Argentina and take more samples. This brings me to the end of my talk. I thank you very much for your attention, and I'm happy to answer any questions about this, and I hope you get something new out of it. Thank you.